Everyone, I'm Anna Lowe, and welcome to Low Oceans, where we submerge and learn. Today's topic is how long-term ecosystem changes affect the biology in the ocean. Specifically, we're looking at multi-decadal shifts between anchovies and sardines off the U.S. West Coast, or if we want to put our oceanography hats on, think about the Eastern Pacific. So starting off, who cares about sardines and anchovies? Aren't they just little fish? Yes. They are both small fish about this big, and they're called forage fish. So forage fish simply refers to their role in the marine food web. So the base of the marine food web are phytoplankton, which are then eaten by zooplankton, which are then eaten by forage fish. Forage fish are then eaten by larger marine animals, ranging from slightly larger fish to seabirds, all the way up to baleen whales. So forage fish are the key link between the plankton, which are basically the size of particles, to larger marine animals that we typically think of when we think about the ocean. So the difference in appearance, or the easiest way to tell sardines and anchovies apart, is that anchovies are a little bit smaller, sardines are a little bit bigger, and they have brown dots along their sides. The big difference between sardines and anchovies really came into focus in the 1930s and 40s, when the sardine population was flourishing, and then all of a sudden it seemed to disappear. John Steinbeck, a popular American author, he wrote about this observation, calling it the sardine regime and its collapse, in his popular novel Cannery Row about fishermen in Monterey Bay. So as fishery science developed, they noticed that the number of sardines being caught by fishermen was fairly synchronous for locations across the Pacific Ocean, from Japan to California down to Peru and Chile. And that the collapse in the 1930s and 40s was the first observation of periods with low numbers of sardines have high numbers of anchovies, and vice versa. So these fluctuations coincide with sardines being most abundant during warmer oceanic periods and anchovies being most abundant during anomalously cooler ocean periods. So from this observed oscillating cycle, scientists drew three conclusions. First, the mechanism for this variability must be fairly consistent or similar in all cases in order to generate this pattern. Second, that pattern was not explainable from fishing pressure, and it's more likely a natural cycle. Third, because the variability is multi-decadal, in this case changing between anchovies and sardines about every 25 or so years, it must be linked to large-scale atmospheric and/or oceanic forcing. So this is a very simplified graphic. Let's dig into some more detailed data. There's a great paper by Chavez et al. In 2003 that examines the sardine-anchovy regime shifts relative to several common long-term records in nature. So all of these records are anomalies, so the increases and decreases are all relative to a long-term average. So starting at the top, we have global air temperature, next the Pacific Decadal Oscillation, then the Atmospheric Circulation Index, the Mauna Loa CO2 record, regime indicator series, and an ecosystem index that's made from seabird counts and harvesting their guano, or poop. The bottom graph shows the time series of seabird abundance, anchovy catch, and sardine catch off the coast of Peru. So you'll notice that the positive anomalies in red and the negative anomalies are in blue. Sections of the atmospheric and oceanic records match up pretty well, and that the changes coincide with the shift between anchovies and sardine catch. So just to reiterate, the warm phases in red coincide with sardines being most abundant, and the cooler phases in blue are when the anchovies are most abundant. So notice in the mid-1990s, we are starting to see a regime shift when the sardine catch plummets from about 4 million metric tons down to just 40,000 metric tons. In the meantime, the anchovy catch is increasing. So one of the ways to extract large-scale spatial patterns in time with multiple variables is through empirical orthogonal function analysis, or simply called EOFs. EOF analysis extracts a spatial pattern from the data and time series of how each pattern oscillates between the negative and positive phases of that pattern. So the top left corner is showing the pattern of the first EOF, or the dominant pattern of sea surface height fluctuations across the entire Pacific. The bottom right, you see the negative phase of the EOF1 pattern, which is called the Eastern Pacific cool phase, because that section of the ocean gets colder, whereas the northwestern and southwestern parts of the Pacific Ocean are getting warmer. During the Eastern Pacific cool phase, the anchovies are most abundant. Then on the top right, you see the Eastern Pacific warm phase conditions, which has the opposite pattern of sea surface temperature. Under these general conditions, the sardines are most abundant. So now we can unpack these ocean conditions in much greater detail and how they impact ocean circulation and other species. So a quick overview of the ocean currents in the North Pacific Ocean. We have the equatorial currents, the subtropical gyre, which rotates clockwise, and the subpolar gyre, which rotates approximately counterclockwise. So let's start with the anchovy regime during the East Pacific cool phase. So focusing on the subtropical gyre, the Kuroshio current off of Japan is slower or weaker on the west side than it normally is, whereas the California current off the US West Coast is stronger. So because the California current is stronger than normal, the thermocline is shallower, which pulls nutrients from below up to the surface by stronger upwelling. These ocean and nutrient conditions propel higher primary production, higher zooplankton, more anchovies, more rockfish, more seabirds, more salmon, but less sardines. Hence, this is called the anchovy regime. Now for the sardine regime during the East Pacific warm phase. So focusing again on the subtropical gyre, the Kuroshio current off of Japan is stronger than normal, and the California current is weaker than normal. So a slower California current causes a deeper thermocline, and therefore there's less upwelling. So weaker upwelling means that less nutrients are being brought up to the surface from below, and there's less primary production. So as a result, off the US West Coast, there's less salmon, less seabirds, less rockfish, less anchovies, but more sardines during this warm phase. So hence these conditions constitute the sardine regime during the East Pacific warm phase. So generally speaking, in the ocean, cooler periods tend to have more primary production and therefore more species abundance, whereas the warmer phases tend to have less primary production overall. And so you see this pattern in ENSO cycles and other types of ocean variability cycles. So there's several positive takeaways from this paper by Chavez et al. In 2003. First, it took over a decade to determine that a regime shift had happened in the 1970s, whereas identifying this regime shift in the 1990s was identified much faster because the oceanographers had learned so much more about the ocean, the atmosphere, and these ecosystem connections. Second, management of ocean resources and studies of anthropogenic effects must account for natural multi-decadal oscillations. Otherwise, the extreme changes in sardine and anchovy abundance, they look alarming in isolation, but they look rather normal when they're viewed with the proper context. And third, identifying a regime shift is much easier than understanding the mechanism behind it. So this paper connected many ocean and atmospheric conditions that seem to oscillate together, but the underlying mechanism that generates these large-scale cascading changes through the system remains obscure. So thank you so much for watching Low Oceans and my brief description of the sardine anchovy regime shifts due to natural long-term ecosystem changes. Please leave any questions about this topic or any other ocean topics you're interested in the comments below. Have a nice day. See you next time.